Everyone. My name is Jyoti Sharma, and this is week 11. What we will cover, let me tell you. So, we will cover from lecture 54 to 58, and in 54 we had pathogen and disease. This was the part 1, and in lecture 55 we have the second part of this pathogen and disease. So, what basically we will talk about is we will talk about pathogen and disease. Then different type of disease we can have. For example, the disease can be spread by the air. So these are the airborne disease. Then the waterborne disease and the food-borne disease. We will talk about this. Then we will go on to epidemiology. So we do have different type of organism that can cause the disease. So we will talk about that. Then we will go to phototrophy. So in which we will discuss a little. We will discuss about the photosynthesis and basically the Kelvin cycle, in which we do have two photosystem, the photosystem 1 and photosystem 2, based on the wavelength that the plant is taking. So we will talk about this, how much ATP has been is taken, and how much ATP is released. So such kind of balance we will try to learn. Then we will talk about the chemolithotrophy. Like the organism may be autotrophic in nature, but in spite of taking inorganic matter, they can take chemicals, for example sulfur. So we will talk about this as well. And then some different specialized bacteria, for example iron-oxidizing bacteria, some microbes, and how the nitrogen cycle is balanced or maintained by the microorganism. So we will try to discuss all these things using MCQs. So I will start my first MCQ uh, with the layer of the skin. So the question is, which layer of the skin acts as the first line of defense against microbial invasion? And we have four options. The first is a subcutaneous layer. The second is dermis. The third is epidermis, and fourth is hypodermis. So you have to give me the one correct option out of these four. You can write in the chat box, even you can speak, and then we will further go on. So Ipsmitha gave me B answer, that this is dermis. Uh, this is actually epidermis, and because this skin has been divided into three parts. The uppermost layer is known as epidermis, and let me show you. Okay, so just see here, the first layer is epidermis. Let me use some pen. So this is epidermis. The first outer layer is epidermis. This is epidermis. Then we have dermis. Then we have subcutaneous tissue, and then we have the muscle. So the outermost layer, this layer is epidermis. So first line of defense is epidermis, and uh, most of the time this epidermis have the dead cell. This layer doesn't have the living cell, but it has the dead cell, and this is the first line of defense. And if it is broken down, then the bacteria can go inside, and that can cause you the infection in the dermis layer, then subcutaneous, and in the muscles. So just keep in mind, this is the outermost layer, this epidermis, and this is the first line for defense, and it is written here as well. The epidermis is the outermost layer of the skin and serves as the physical barrier to block pathogen from entering the body. It is made up of tightly packed cell and include keratin. So keratin is the one of the protein that in our hair also, that protein exists, and which further do the protection. So just keep in mind, for the skin we have basically three layer. The outermost layer is epidermis, then dermis, and then subcutaneous tissue. And that's why this epidermis is the right answer, because this is the first line of defense. Is it clear, Ipsmitha, why this is epidermis but not the dermis? Yes. Okay. Thank you so much for responding. Okay. The next question is, which enzyme found in human sweat is responsible for breaking down bacterial cell walls? So we have four options: lactase, amylase, lysozyme, and protease. Which answer you think right? You can write in the chat box out of this A, B, C, D. So you have to tell me the name that is found in the in our sweating. This should be lysozyme. Okay, let me tell you. Like initially in, I think 2 or 3 weeks ago, I told you the nomenclature of the enzyme, in which we have ASE at the end of any enzyme. So this ASE is like common to the to any name of the enzyme, but first is the name of the substrate. So lactase means the substrate is lactose, and that will, let's say, breaking down of the lactose with the help of enzyme lactase. So this is lactase. Amylase, so you have one substrate, amylose. So if you are going to break down the substrate amylose, then the enzyme will be used is amylase. But here, because we won't so for example, this is our skin and this is bacteria. Okay, and if we do have sweating on the skin, then this sweating have some enzyme. What that enzyme will do, that enzyme will break down the cell wall of this enzyme. Okay, so breaking down means lysis, right? Breakdown means lysis. So based on that, if we do have some enzyme that is breaking down the cell wall of the bacteria, then that enzyme name will be lysozyme. Okay, so this lyso originated from lysis. So lysis means breaking down, and this breaking down is of bacterial cell wall. And if the bacterial cell wall is getting breaking down, it means the the bacterial cell is not going to infect the human body or the human skin. So we have a very powerful and protective enzyme that is known as the lysozyme in our sweating. So whenever we have the sweating, that enzyme is secreted, and that enzyme help us to protect from the bacterial infection. So, and the name is taken from the lysis. So lysis means breaking down. So the enzyme name is lysozyme. So that's why C is the right answer. Okay. I think I have cleared it. But just see here. So lysozyme is an antimicrobial enzyme found in body fluid like tears, and it is not just present in the sweating, but it is also present in the tears and in the saliva and sweat. It attacks the peptidoglycan, as we already have talked about this. The cell wall of the bacteria have one compound, that is peptidoglycan layer. So it attacks the peptidoglycan in bacterial cell wall, and that's why it will destroy many type of the bacteria. So that's why sweating also protects us from the bacterial infection. And so if you can see here, in the ear we have tears and we do blink, and we do have lysozyme in the tear that will help us from the bacterial infection. If we are talking about the skin, then we already have talked that skin have the epidermis, that will epidermis means dead cell. We do have that dead cell will protect us from any kind of infection. And we do have lysozyme in the sweating, but we also have lactic acid, propanoic acid, and some normal flora that can protect us from any of the bacterial infection. And likewise, in respiratory tract, in gastrointestinal tract, and even in urogenital tract, we do have some enzyme and some compound that can help us to protect from the bacterial infection. But the question was just in the sweating, which kind of enzyme is found. So the enzyme is lysozyme. Okay, so coming to the next question, we already have talked about epidermis, we talked about the lysozyme, and this question is basically very similar to the last two questions. So you just need to match the skin component with its correct function. And so we have epidermis, we have lysosome, and you have to tell the function. So the functions are: breaks down microbial cell wall, and B is provide a physical barrier against pathogen. And you need to match the skin component with its function. So we have A, B, C, D. Can you tell me the right answer for this? Epshmita gave me answer B. Anyone else? Anyone else wants to give me the answer? Okay, so yes, the answer is B. We already have talked about this. That the work of epidermis is that it will provide the physical barrier against pathogen, and this is the topmost layer of our skin. And in the sweating, we have the enzyme that is known as the lysosome. So lysosome will break down the microbial cell wall, and this is how it can protect us from the bacterial infection. So, like the likewise you can give me the answer, so the answer will be B. Okay, coming to the next question, but before that, you can read here. The epidermis act as the first physical barrier, while lysosome chemically destroy microbial. Both are the part of innate immune system. So innate immune system means the already immune system that we have, but function different, one structurally and other enzymatically. So epidermis is a kind of structure, so it is providing us the protection structurally, but lysosome is the enzyme, so it is protecting us from bacterial infection enzymatically. Okay, now which of the following bacteria is correctly matched with the plague type it can cause? So we have the name of the bacteria, and in the right side we have the type of plague it can spread, and you have to tell me the correctly matched. And we have four options. So we have four bacteria: Streptococcus mutans, Yersinia pestis, and Prevotella intermedia, and then Porphyromonas species. And we have four plague. So likewise, you can tell me the answer. Which one is the correct one? Any guess about this? So just keep in mind this Streptococcus, this Prevotella, and this Porphyromonas. These are the bacteria that we have in our mouth. Mostly, they can cause some gum problem, and these are not causing the plague. But this bacteria, Yersinia pestis, this can cause the plague. So that's why this is the right answer. And the bacterias, this A, C, D, these are not the bacteria that will cause the plague, but these are found in the mouth. So that's why B is the right answer. And I also have some explanation here. And this is causing the bubonic plague. So this is the causative agent of the plague, which appear in three main clinical forms. So this single bacteria can cause three different type of plague. So this can be bubonic, this can be septicemic, and the pneumonic. And the other three, as I told you, these are associated with the oral or dental infection, not somatic disease like plague. Okay, so so I think this is clear. Okay, again, the question is like that only. As I told you, the Yersinia pestis is the main bacteria that can cause the plague, but we do have some oral pathogen, for example, Streptococcus mutans and the Porphyromonas, and you just have to tell why these bacteria have some differences. So, which statement correctly distinguishes Yersinia pestis from typical oral pathogens like Streptococcus mutans and Porphyromonas species? So we have four options. Yersinia pestis forms spores to survive in harsh environment. The B is oral pathogen are motile, while Yersinia pestis is non-motile. Then third option is Yersinia pestis is transmitted via saliva, while all pathogens are flea-borne. And D is Yersinia pestis causes localized gum infection, unlike oral bacteria. So, what do you think? What is the right answer for this? How the plague causing bacteria is different from the oral pathogens? Not exactly C. Actually, the answer should be B, because oral pathogens are motile in nature, so they can move. Bacteria can move from one place to another place, but here Yersinia pestis is non-motile in nature. It can't move, so there is no any kind of movement of this bacteria. So that's why B is the right answer. Yes, so let me show you the explanation here. Yersinia pestis is non-motile, while some oral pathogen, Porphyromonas, show limited motility due to fimbria or gliding movement. But the option that is given, that are showing you the motile nature. Okay, the next question is. So again, this question is based on Yersinia pestis only. So Yersinia pestis is primarily transmitted through respiratory droplet. This is the assertion, and the reasoning is it causes ulcerative gingivitis in oral cavity, and you have to tell uh, A, B, C, D. So in A, both A and R are true, and R is the correct explanation of A. In option B, both A and R are true, but R is not the correct explanation of A. In C, A is true, but R is false. And in D, A is false and R is also false. So you have to tell based on the assertion and reasoning that is provided. So what do you think? What is the right answer for this? Okay, so the answer is D, because we already have talked about that. Uh, that uh, this Yersinia pestis is one of the bacteria that can cause the plague, and this plague is through the rats or through the fleas. So this is not the right assertion. Then if we are coming to the reasoning, then this is causing the plague, but not any problem in the oral. So that's why reasoning is also false. So that's why D is the right answer, that A is false and R is also false. Here is the explanation that I have given. So Yersinia pestis is primarily transmitted via flea bites, not respiratory droplets. And that's why A is the wrong statement or wrong assertion. And it does not cause ulcerative gingivitis. That's due to oral bacteria like Prevotella intermedia. So this both assertion and reasoning were false. I think this is clear. Okay, so coming to the next question, uh, what is the primary reason for saline solution are administered to severely ill patient instead of solid food? So we have four options. Pathogen required complex food to survive, so removing them starves the bacteria. uh, B option is for saline enhance nutrient absorption from complex food. C is for saline bypass the impaired digestive system and prevent dehydration. And four is saline neutralizes the enzyme secreted by pathogen in the gut. So the question is very simple. Whenever we get ill, or we do have like fever or something else, then we go for the saline solution, so and directly saline solution using the syringe. So the question is, why do we need this saline solution? What is the need? Why we can't take the solid food? Instead of solid food, we need the saline solution, but why? Can you tell me the answer for this? Why do we need, or you can take the example of, let's say, glucose. So if someone have is taking glucose using that drip and using injection, so why this is the case? Why the patient cannot eat the solid food, but why it is taking saline solution? You are confused between C and D. Okay, D is not the right answer. Saline neutralizes the enzyme secreted by pathogen. No, saline is not neutralizing the enzyme. So the remaining option will be right, Smita. So yes, C is the right answer. And the reason is very simple, that in the gut initially you have 100 or 1,000 type of bacterias in your stomach, in your intestine, that can help you to digest the food. But whenever you get ill, you take the antibiotic, so that the work of that antibiotic is to kill the bacteria. So the this antibiotic not only kills the bacteria that are harmful to us, but it will also kill the bacteria that are useful for us, useful for the digestion of the food. So based on that, if you are taking any kind of antibiotic, so it is killing the beneficial bacteria that you have in the stomach or intestine. So, and that bacteria are useful for the digestion. So at that time, you, because you do not have sufficient bacteria in your stomach that will digest the food, so the food will not be digested by itself, so you can't take the solid food. So that's why it is very much advisable that you just take the saline solution, so that you can bypass this disturbed digestive system, and that can prevent us from the dehydration. So I think this is the like clear explanation that I tried to give you. Just simple is that the bacterias, the useful bacterias are killed by the administration of antibiotic, and that's why the digestion cannot be completed without that useful bacterias. So that's why we have to bypass this defective or impaired digestive system. So just see here how many like good bacteria. So these good bacteria are mainly referred as probiotics. So these probiotics may have lactobacillus. Lactobacillus is a very good probiotic that can help you in the digestion. Then we have lactococcus, we have propionibacterium, we have streptococcus thermophilus, and more. So we have a lot of bacteria in our stomach, and even the fungi that can help us for the digestion. And in the presence of antibiotic, if we are taking, these bacteria are killed, and that's why we have to take saline solution in spite of taking the solid food, so that we can have some energy, we can avoid the dehydration. And we have a lot of like good bacteria, so you can read more about these bacteria. Okay, so here is the right answer that you can read. In explanation, in severe infection, the gut's ability to process nutrient is compromised due to disruption of the normal microbiota, and this four saline provide fluid and essential electrolyte without taxing the digestive system, and that will help to maintain the hydration and the basic homeostasis. Okay, the next question is, which of the following best explains how pathogen infection affects nutrient absorption in the intestine? And we have four options. Pathogen increase surface area of villi, reducing nutrient flow. And B option is pathogens destroy beneficial microbe that assist in digestion. C is pathogen block digestive enzyme from reaching the liver, and D is pathogen binds to glucose and make it unusable. So, how pathogen infection affects nutrient absorption in the intestine? The explanation I already have given in the last question, so I think from that you can give me the answer for this question. Anybody? Anybody wants to give me the answer for this question? How pathogen infection affects nutrient absorption in the intestines? Very simple explanation, that because pathogen destroy beneficial microbe that assist in digestion. As I told you, whenever we do have the pathogen infection, that pathogen infection, and we have to take the antibiotic, that antibiotic will kill the beneficial microbes, and this pathogen will also give the competition to the beneficial microbes, and this beneficial microbes have to be killed in that competition, because these pathogen infective bacteria are very opportunistic in nature, and that's why whenever you do have any kind of infection due to pathogen, so that will destroy your good bacteria or microbes in your stomach, in intestine, that help us in the digestion. So this is how any infection will affect nutrient absorption in the intestine. Okay, so explanation is the normal gut flora helps break down food and synthesize certain vitamins. When pathogens invade, they can outcompete or kill this beneficial bacteria, and this is how this will disrupt digestion and nutrient absorption, especially for complex foods. Okay, so the next question is, we have one assertion, and this assertion is, a sick person may be unable to digest fibrous or high fat meals, and reason is pathogen induced disruption in gut flora compromise enzymatic breakdown of the complex nutrient. And you have to tell me out of these four options, and in the first, both A and R are true, and R is the correct explanation of A. In B, both A and R are true, but R is not the correct explanation of A. And in C, A is true, but R is false. And in D, A is false, but R is true. Let me see who has given me the answer. Ipsmita has given me answer A, and yes, I agree with this question and even your answer, that uh, yes, this is true, that a sick person may not able to digest a complex food, for example, the food that have high fat. And why this is because And this pathogen can disrupt the gut flora. We already have talked about this. So that's why A is the right answer. So the explanation here is the gut flora plays a key role in breaking down tough components like fiber and fats. When illness disrupts this microbiome, the body struggles to digest such foods. Hence, simple foods or fluid are recommended until recovery. And you already have given the right answer. So let me go to the next question. Okay, so we do have another bacteria, and the name is Klebsiella pneumonia. And the question is, why is nitrogen fixation by um, Klebsiella pneumonia particularly important for the diet of the person that persons are in pneumonia? And we have four options. Their food is rich in nitrogen but poor in protein. B is their diet lacks sufficient nitrogen source due to low protein intake. C is they consume too much animal protein requiring nitrogen recycling. And D is K pneumonia digest sweet potatoes for them. So this bacteria, Klebsiella, so this Klebsiella bacteria is very useful for the person that persons are located in the country or the place. So this is a pneumonia. So you just have to tell me why this is very important, that bacteria to have in their stomach. So if you people know that this New Guinea is a place that is tropical in nature, and in tropical region, the staple food is mostly potato-like thing. So you have seen the yam or some potato-like sources. So that energy sources are mostly carbohydrates, right? So what will happen? So in the tropical region, when there is a ample amount of potato or the carbohydrate source, that place do not have the protein source. Okay. And because we need nitrogen, and that nitrogen will help us to synthesize the protein. So in human we need the nitrogen, but how we can fix that nitrogen in our body? So because at that place do not have the protein rich food, but one bacteria that is present in their gut will help to fix that bacteria, and if sorry, to fix that nitrogen. And if the nitrogen will be fixed by that bacteria, then this nitrogen can be utilized to synthesize the protein. So at that place we do not need to have the external source of protein as in the form of food, but that bacteria will help to fix the nitrogen and to give like this will help to synthesize the protein within the body. So this is how this Klebsiella pneumonia bacteria can help to the people of pneumonia. So the answer will be that that place, the tropical place, in that diet lacks sufficient nitrogen source due to low protein intake, because at that place there is like the ample of carbohydrate source but very low protein food they have. So that's why they do not have the nitrogen source. So this is kind of adaptation or god gift to that people, that they also they have that bacteria in their gut, and that bacteria will help to fix the nitrogen. So that's why B is the right answer. Here I have given the explanation. New Guinean diets consist mainly of sweet potato, which are high in carbohydrate but low in protein. And K. pneumonia, being a nitrogen fixing bacterium in their intestine, compensates by converting atmospheric nitrogen into usable forms, and that's how it will support protein synthesis and the nutritional needs. So just keep in mind, in tropical countries, there is ample amount of carbohydrate source, but they do not have protein source, and that's why they do not have sufficient nitrogen in the body. So but in their body they have the bacteria, and that is the Klebsiella pneumonia. That bacteria can fix this nitrogen from atmosphere into their body, and that can help them to synthesize the protein in their body. So that's why the B is the right answer. Okay, which of the following bacteria is not known for nitrogen fixation and is unrelated to compensating low protein diet? So, along with, because we already have talked about Klebsiella pneumonia, and along with that, there are a lot of more bacteria that can help us for the nitrogen fixation. And here the question is the bacteria which is not known as, not known for nitrogen. So the options are Klebsiella pneumonia, B is Azotobacter, C is Streptococcus, and D is Rhizobium. What do you think? What is the right answer for this? Ipshmita gave me answer C, and I also agree with C. Is anyone giving me the contradictory answer for this? Or everyone should go for option C only? Okay, so yes, C is the right answer, because we already have talked about the Klebsiella, because this can fix this atmospheric nitrogen. But along with that, you already have learned that in the legume roots. So, for example, if there is any pulse or peas in the root of that legume, we have some bacteria that can also help for the nitrogen fixation, and that bacteria are, some of the name of that bacteria are Azotobacter, Rhizobium, right? So just see here, and there is a classification of this nitrogen fixation bacteria. So they can be so nitrogen fixation can be done by microorganism and by some other factor. If it is nitrogen is being fixed by abiotic factor or not living organism, then this will come under abiotic. So for example, whenever there is a thunderstorm or there is any electric discharge, that can also fix the nitrogen. So this will come under the abiotic. And even by like some artificial chemical reaction, you can fix the nitrogen. But if we are talking about the nitrogen fixation by microorganism, so this has been classified into two parts, free living and symbiotic. So free living means bacteria or any microorganism do not need to have any relationship with another organism. So for example, and okay, so this is free living, so they are independent of any another organism. And if they do have some relationship with some other another organism, then this will come under symbiotic. So for example, we have talked about rhizobium. So rhizobium needs a relationship with the root nodule of any legume plant. So so there is a kind of relationship between legume plant and the bacteria. So this will come under symbiotic. But if they can fix the nitrogen without taking help of any another organism, so this will come under free living. So free living can be, for example, Azotobacter, Rhodospirillum, and then Nostoc. But if you are talking about the bacteria but that have the symbiotic relationship, so example of that, these can be ribozobium, azoribozobium, right? And just see here the explanation. So Streptococcus pneumonia is a pathogen causing pneumonia and other respiratory illness, but they are not involved in the nitrogen fixation. And another that we already have talked about, Klebsiella, Azotobacter, Rhizobium are well-known nitrogen-fixing bacteria. And the last one, particularly in plant root nodules. So we already have talked. So just see here, ribosobium and the rhizobium, these have the symbiotic relationship with the root of the legume plants, or even with the stem nodules. Okay, so the next question is, nitrogen fixing gut bacteria can improve survival on carbohydrate rich low protein diet. So this is the assertion. The reason is these bacteria convert atmospheric nitrogen into biologically available nitrogen compound, which can be used for amino acid synthesis. And based on this assertion and reasoning, you have to tell me the right answer out of this A, B, C, D. Why the answer is not A? If Smita, you have given me the B answer that R is not the correct explanation of A. As we already have talked that if we do not have protein rich diet, then some bacteria can help us to fix the nitrogen, and this nitrogen can synthesize the protein in our body. So why this R is not the correct explanation of A, because you have given me the option B as the right one. Okay, so A is the right answer because I already have talked about this, right? Uh, in tropical countries, we do have the carbohydrate rich food, but they do not have nitrogen in their food, and that's why they do not have protein. And like the food is protein deficit. So, and that's why some bacteria, for example, Klebsiella pneumonia, help to fix the nitrogen, and this nitrogen is the integral part of the amino acid, and this amino acid is again the integral part of the protein. Okay, so if we are able to fix the nitrogen in our body, then this nitrogen can be used to synthesize the protein. So even if we do not taking the protein rich food, even then we can synthesize the protein in our body. Okay, because we do have some gut bacteria that can fix the nitrogen. So that's why A is the right answer, that both the statement are correct, and R is the right explanation of A. And just see here, in low protein diet, nitrogen is often limiting. Gut bacteria like Klebsiella pneumonia provide biologically accessible nitrogen via nitrogen fixation, enabling the host to synthesize essential amino acid, thereby supporting nutritional balance and survival. So I think this is clear now why the answer is A but not B. Okay, thank you. Next question is based on another bacteria, and this is Proteus species. And the question is, what is the main biochemical mechanism by which Proteus species alter the urinary environment during infection? And option are production of lipase that increase urine acidity. B is hydrolysis of urea to ammonia, increasing urine pH. C is the production of lactic acid to suppress host immunity. And D is breakdown of glucose to ethanol in the urinary tract. So this is the one species that can disrupt the urinary environment, and that can cause the infection. And how it can cause the infection? You gave me a B option, and yes, I agree with the B option, that it will do the hydrolysis of urea, so it will change urea to ammonia. And whenever there is conversion of urea to ammonia, that this means urea is very acidic in nature and ammonia is not. Ammonia is alkaline in nature, is basic in nature. So if from we are converting urea to ammonia, means we are changing the pH from acidic to basic. So if it is going from acidic to basic, means we are increasing the urine pH. And we already have seen in one of the schematic diagram that uh, the pH of the urine is also one of the defense system of our body to protect us from the bacterial infection. And if some species is altering the pH of your urine, it means this can give you the infection, right? So B is the right answer. Hydrolysis of urea to ammonia, increasing urine pH. And just see here the explanation. Protease species produce urease, so it secretes the enzyme. That enzyme is known as urease, which catalyze the breakdown of urea to ammonia. This result in alkalization of urine, creating an environment favorable for bacterial survival, and contributing to stone formation and upper UTI complications. So just keep in mind, if there is some bacteria and that bacteria are changing the pH of your urine and like converting it from urea to ammonia, it means it is providing the conducive environment for another organism to survive, another bacteria, fungus to survive at that part, and that can give you the further infection, right? Okay, coming to the next question. Clear this. Which of the following correctly describes protease mirabilis? And options are gram positive aerobic cocci, B is gram negative facultative anaerobic rod shape. C is gram negative obligate anaerobic spore forming. And D is gram positive facultative anaerobic rod shape. So you just have to tell the characteristic of the bacteria, and that bacteria is Proteus mirabilis. You have to just tell me either it is gram positive or gram negative. And we already have discussed that this gram positive and gram negative is based on the cell wall. And then it is aerobic or anaerobic in nature. So while respiration, it is taking oxygen or not. And if it is taking oxygen, whether it is mandatory to take the oxygen, or there is no any mandate to take oxygen every time. So it can be facultative or obligate. Obligate means it is obligatory. Obligate anaerobic means that it is obligate or necessary to do not have the oxygen presence. Facultative anaerobic means if there is no any oxygen, then that bacteria can act as anaerobic. So obligate means obligatory, and facultative means if there is not any presence of let's say oxygen, that it can act as anaerobic in nature. And the third is the shape of the bacteria, either it is cocci or the rod shape or spore forming or rod shape. So you just have to tell me the characteristic of this bacteria, Proteus mirabilis. So the answer is B. So this is gram negative. So as and we already have talked about gram negative bacteria, how this gram negative bacteria is different from gram positive, and how we can do the staining, and based on that staining how we can distinguish whether our bacteria is gram negative or gram positive. We already have talked about this. Then facultative anaerobic means, in the presence of in the absence of oxygen it can act as the anaerobic, and but if there is ample amount of oxygen, that it will act as the aerobic in nature. So that's why this is facultative anaerobic, and these are rod shaped bacteria. So B is the right answer. Okay, the next question is, how do urease? We already have talked that this bacteria uh, produces the enzyme. This enzyme is known as the urease. So the question is, how does urease production by protease species contribute to kidney damage? And four option are: urease binds to nephrons, blocking urine flow. B is urease causes inflammation of the liver, affecting kidney metabolism. C is ammonia produced from urea raises urine pH, promoting infection and stone formation. And D is urease paralyzes smooth muscle in the bladder. If Smita gave me answer C, and yes, I agree with C. We already have discussed this. So I don't think that I should explain much about this. Just read here the explanation. Urease convert urea to ammonia, which alkalized urine. This high pH can lead to the formation of some stone that will irritate the urinary tract, and that will facilitate bacterial colonization. Thereby, it will increase the risk of some disease. And so this is how it can affect the kidney. Okay, so coming to the next question. Okay, which of the following disease is least likely to cause a host-to-host pandemic, endemic, sorry. And we have four options: COVID-19, influenza, cholera, and measles. So the question is, which is least likely to cause a host-to-host epidemic? So we already have talked. What do you mean by host-to-host pandemic? So host-to-host pandemic. So whenever we do have the sneezing, so there is a direct connection. Like we are transferring the bacteria, the microbe from one human to another human. So we were initially the host of the bacteria, and or let's say the virus, and we are transferring that virus or bacteria to the another organism or another human. So this is known as host to host pandemic. And the question is, which disease is least likely to cause a host to host pandemic? And Smita gave me some answer. Let me see which. So she gave me C answer, and yes, I will also go with the cholera, because you people know that cholera is caused when your water or food is contaminated. Then only if a human or host will take that food, then it will get infected. But this is not host to host. So for example, if one person has the cholera, then another person will not have the cholera until the infected person will secrete the feces, and this feces will contaminate the food or the water source, and that again the water source and the food source will cause the infection and will give you the cholera. So this is the circle of the cholera, but this is not like host to host. If we are sneezing and if we are transferring the virus in the air, and that the air is taken by another host, so this is how this is host to host, but cholera is not one of that part. So just see here, like this is just the schematic diagram, that is like whenever we have some agent or the microbe that can cause you the infection, and then we have the environment, and that environment can act as either the medium or even the carrier of this agent, and then the host will get that agent. So, cholera is infectious. It is primarily spread through contaminated food or water, not direct person to person contact. It causes common source epidemic rather than host to host pandemic, like influenza, COVID, measles, which spread via respiratory droplet or close contact. Okay, the next question is, which characteristics best defines a host to host endemic? And the option are rapid spike and decline in case due to contaminated water resource or water source. B is gradual rise in cases due to secondary transmission between individual. C is occurrence limited to animal before jumping to human, and it is spread only through non-living vectors like fomites. So you have to tell me the characteristics of the host-to-host pandemic or endemic. Okay. Yes. So you gave me answer B, and yes, this is the right. So, as I told you, some epidemic are not host to host, but depend on the vector, and even some are uh, waterborne. So, and that waterborne are the disease that give you the rapid spike in the infection. But if it is a host to host, then this will give you the gradual rise in cases. But if there is the infection is very fast, for example in COVID-19, then that can give you the rapid spike as well. So just see here, host to host epidemics typically show a slow progressive increase in case as the disease is passed from one infected person to another, unlike common source epidemics like cholera, which cause a sharp rise and fall due to single exposure source. So for example, there is a water source, for example a well, and in that well you have contaminated water, and thousands of people are extracting that water for the use. And so that's why whenever a person will drink that well water, this will give the infection to thousand of the people. But and that's why there is a sharp rise and fall due to common source epidemic. But if we are talking about host to host, then this is always slow and progressive. Okay, so next question is, which of the following would most likely be controlled by isolating infected individual rather than improving water sanitation? And option is typhoid, influenza, cholera, hepatitis A. So, which of the disease we can control by isolating the infected individual rather than improving water sanitation? It's me the gave me answer D, and this is influenza. So as I told you, now you just have to see the difference between disease that are host to host disease and the one is common source disease. If this is host to host, so if you are isolating one host from another, that's how it can resist the infection in another host. But if it is a common source, so for example, as I gave you example of well, so if that well have the contaminated water. So like if we will improve the sanitation, that's how we can improve the we can resist from the cholera. But because here the question was most likely be controlled by isolating infected individuals, so this is the best example what we did in COVID-19. If some person have some if that person had the COVID-19, then what we did is we isolated that person so that that person do not have any contact with another person, and that person will not get infected from the infected person, right? So this is how we can control if that disease is host to host disease. And here the influenza is the host to host disease. Influenza is such like if you have any cold, right? So in if you have any cold or cough, then no, because this is the respiratory droplets. And if we are not like if we are doing this sneezing in a separate room, then this is how we can avoid the infection. So that's why influenza is the right answer. So influenza spread by respiratory droplet, making isolation and respiratory precautions key to controlling its spread. On other hand, typhoid, cholera, and hepatitis A are largely water or food-borne, and that requires sanitation and hygiene improvement to prevent outbreaks. So that's why B is the right answer, but not A, C, and D. Okay, so now we will talk about some career of the disease, and you just have to tell me which mosquito genus is correctly matched with the disease it transmit. So these are the some of the genus of the mosquito, and in the right hand side we do have the disease name, and the option are Aedes and malaria, Culex, dengue, Anopheles, malaria, Culicita, yellow fever. So you have to tell me which is the right matched, which mosquito is right matched with the disease it transmits. Anopheles is not giving you malaria. This is a very common example, like a lot of like everywhere we get that mosquito is giving you the malaria, and which mosquito is like giving that malaria, this is anopheles, right? So C is the right answer, that for the malaria we need a particular mosquito genus, and that genus name is anopheles, and that's why C is the right answer. And here in the explanation you can see only the female Anopheles mosquito transmit the malaria parasite. The name of that malaria parasite is Plasmodium. So mosquito is is not giving you the disease directly, but there is one parasite or microbe on the mosquito. So mosquito is just the carrier of that parasite, and that parasite name is Plasmodium. So you have Plasmodium. If that Plasmodium is on the mosquito, and that mosquito should be female Anopheles, then only it can give you the malaria. And Aedes, so if we are talking about the Aedes genus of the mosquito, that will transmit dengue, chikungunya, Zika, and yellow fever. If we are talking about the Culex mosquito genus, then this is linked with the Japanese encephalocytis and West Nile virus, but not the malaria. So this is how we can correctly match the mosquito genus with the disease it transmits. And here you can see this is one of the important schematic diagram that you can see. So initially what uh, we have the mosquito, right? And mosquito, in the gut of that mosquito we do have the plasmodium, plasmodium parasite. So this is Plasmodium parasite that can give you the malaria. So, firstly, in the mosquito midgut, we do have the Plasmodium. Then it will create oocytes. So, oocyte is a kind of female reproductive spores, you can say. So, it can produce some spores. That plasmodium inside the mosquito, then this will give you the sporocyte. This sporocyte um, are injected by the biting. So whenever the mosquito will bite the human, then it will give you the sporocyte in your human body. Okay, so the sporocyte of the plasmodium will be given by the mosquito to the human body. Okay, and what will happen, this porocyte will penetrate into the liver cell, then they will mature, then they will be converted into merocytes, and then it will produce as the gamocyte, and this is how this gamocyte will again taken up by the mosquito uh, from the feces and all. So this is how there is a cycle, that mosquito is just the carrier of the infective agent, and that agent is plasmodium. Okay, so the next question is, in the context of malaria transmission, which of the following statement is accurate? And we have four options. The malaria parasite uses only mosquito as its host. The B option is mosquito acts only as a passive carrier of the parasite. C is both human and Anopheles mosquitoes are host for the malaria parasite, and D is in the life cycle. We already have talked about this in the last figure, and based on that, you can tell which is the following accurate statement. We already have talked, right, that plasmodium parasite is not having the host, that is the Anopheles mosquito, but the host is also human. So that malaria parasite have both human and the Anopheles mosquito that will act as a host for the malaria parasite. We already have seen, right, this one, or so here, I think this figure will give you better. So this plasmodium spores, these are the plasmodium spores. So these plasmodium spores are not only present in the mosquito, but they are also present in the human. So this plasmodium parasite has two hosts, the one is mosquito and another is human. So these parasite have two hosts. So that's why if we are giving the answer of this, then the answer will be both humans and Anopheles mosquitoes are host for the malaria parasite. And here again, you can see in the explanation, the malaria parasite, Plasmodium species, undergo different stages in its life cycle in both the mosquito and human host. The mosquito is the definitive host where sexual reproduction occur, and human is the intermediate host where asexual reproduction occurs. So that's how you can give me the answer. C is the right one. Okay. So next question is, which of the following best define a disease vector in epidemiology? And the options are a genetic mutation that enhance infection. B is any microbe that can multiply within a human host. C is a non-living surface that help transmit disease. And D is an organism that transmits a pathogen from one host to another. So you have to define what do you mean by vector or disease vector. How you will define like this is the disease vector? Ipsmita gave me the answer that an organism that transmits a pathogen from one host to another. And yes, I agree with this option. So this is just like that mosquito. So whenever I'm saying that mosquito is just the carrier of any parasite that will give a human infection, then so this carrier are just the disease vector, so that will transfer or transmit a pathogen from one host to another host. So for example, we have a lot of bacteria that are present in, let's say, in buffalo or cow. And if that buffalo and cow uh, like are the carrier of this disease and will give that disease to the human, then this cow or buffalo may be considered as the vector. And for example, if uh, we have the fruit flies, right? We have mosquito, we have a lot of flies, and that flies can be act as the carrier of any disease, because that are carrying some bacteria or let's say virus that can infect the human. So they are known as the disease vector. So any organism that transmit a pathogen from one host to another. So here you can see the explanation. A vector refers to the living organism, often an insect or arthropod, that carries and transmit an infectious pathogen. So, for example, in malaria, Anopheles mosquito is a vector, while Plasmodium is the causative pathogen. And you can just see here a mosquito, biting fly, flea, mite, and ticks, house fly, cockroach. So these can be vector. Here again you can see so these insects have some virus, and these viruses are causative agent, but these are just the vectors because they are carrying this bacteria. Okay, the next question is, which of the following action would be most effective in reducing the spread of typhoid fever? And if you need to like reduce the spread of typhoid fever, then what action would be most effective? Options are using insect repellent in tropical region. B is avoiding raw vegetable in cold climate. C is practicing good hand hygiene after using the toilet. And D is wearing mask in crowded public areas. First, you have to see typhoid fever is host to host or common source epidemic, and based on that you can give the answer for this, whether we need the isolation or we need the hygiene condition to control this spread of the typhoid fever. Yes, I agree with your answer, and your answer is C. So, practicing good hand hygiene after using the toilet, as we know that if someone have the typhoid, the another will not have the typhoid, because this is not the host to host epidemic, but this is due to the contaminated food and the contaminated source. Or for example, in the feces we have this kind of bacteria that can cause the typhoid. So if we are washing our hand after the toilets, so that's how we can avoid that bacteria to get contact with the human body, and this is how we can reduce the spread of typhoid fever. So just see here, this is the feces, and in that feces we have like a lot of that bacteria that can give us the typhoid. And if whenever we are washing our hand after like using the toilet, then we can avoid any bacteria to be in our food or in our mouth, so that we can avoid the typhoid. So just see here the explanation. Since typhoid is primarily transmitted by the fecal oral route, proper health hygiene, especially after using bathroom and before handling food, plays a critical role in breaking the chain of transmission. Okay, so coming to the next question, which of the following pathogen can cause typhoid or typhoid-like symptoms? So we have four options: Escherichia coli and Salmonella enterica. The next option is Salmonella typhi and Salmonella paratyphi. C is Vibrio cholerae and Shigella flexneri, and D is Salmonella typhimurium and Yersinia pestis. So these are the name of the bacteria, and you have to tell me what bacteria can cause typhoid or typhoid-like symptoms. If Smita gave me B answer, and again I agree with this B answer, that this is the Salmonella typhi that will give you the typhoid, and Salmonella paratyphi, this will give you the typhoid, and that's how this will give you the typhoid-like symptom, because this is not exactly the typhoid, but this is the paratyphoid, and that's why B is the right answer. And just see here the explanation here. Typhoid fever is typhoid fever is caused by Salmonella enterica, serover typhi and paratyphi. These bacteria are transmitted through transmitted food, contaminated food, and water. Another bacteria like Escherichia coli or Shigella can cause gastroenteritis, but not typhoid fever. So that's how you can give the answer. And here I also provided you the difference between Salmonella typhi and Salmonella paratyphi. And just see here that Salmonella typhi is the agent that is responsible for causing typhoid fever, and another is responsible for causing paratyphoid fever. Salmonella typhi is highly prevalent, and this Salmonella paratyphi is not as prevalent as Salmonella typhi. So this is the difference between Salmonella typhi and paratyphi. Okay, so the next question is, which type of disease transmission best describe how typhoid spread? And the options are zoonotic and airborne, B is food-borne and water-borne, C is vector-borne and droplet, and D is fomite-based and sexually transmitted. So, how typhoid is spread? So yes, we already have talked about this, that typhoid is due to the contaminated food and water. So typhoid is mainly food-borne or water-borne disease. So that's why yes, B is the right answer. And just see here, typhoid is not transmitted by animal or vector, but through ingestion of food or water contaminated by human feces containing Salmonella bacteria. Hence, it is a food or waterborne, not vector or droplet borne. So that's why B is the right answer. Okay. The next question is based on the difference between pathogenicity and virulence. So we have four options. Pathogenicity refers to the shape of the pathogen. Virulence is its reproductive rate. B is the pathogenicity is the ability to evade the immune system. Virulence is the ability to replicate. C is the pathogenicity refers to the ability to cause disease. Virulence refer to the severity of that disease. D is pathogenicity is caused by antibiotic and virulence is caused by toxins. So you have to tell me the difference between pathogenicity and virulence. What is the difference between these two terms? Okay, so you think that C is the right answer, and yes, your answer is right. That pathogenicity is the ability to cause a disease, and this is the microbe. This is the property of microbe. Based on that, pathogenicity will be defined. But the virulence is the severity of that disease, and I think in next question we will clarify this. Just see here again, the pathogenicity is the broad capacity of microorganism to cause disease, while virulence specially measure how severe or damaging the disease is. For instance, both the common cold virus and Ebola are pathogenic, but Ebola is more virulent. And here again, you can see the difference between these two. So virulence is the measurement of the ability to cause a disease in the host organism. Pathogenicity is the ability to cause a disease in host organism. And you can read more about the difference between these two. But I think this is clear what's the difference between virulence and the pathogenicity. Okay, next is which of the following could be considered a virulence factor in a pathogen? And the options are presence of the rigid cell wall, B is the production of enzyme that break down host tissue, C is the ability to float in air for long period, and D is a slow reproductive cycle. So, like for the pathogen do have a multiple virulence factor that can give you the disease. And like why? What are the reason that it can give you the disease? They do have some advantages. So based on that, that advantages will come under the virulence factor that they have in the pathogen. So you have to tell which one is considered a virulence factor in pathogen. What that pathogen do have that have the advantage that it can give you the some infection. Not exactly C, but you can rely on answer B, that this is the production of enzyme. As I told you, like for the pathogen to give the infection, bacteria or the virus have to break down the host tissue. Then only it can get penetrated in the host body, and that's how it can produce the infection. So if some pathogen have the enzyme that can break down the host tissue, so this is the one of the factor that it is a virulence in nature. And we do have a lot of like factor. I will tell about this. So just see here, virulence factor are the component or the trait that enable a pathogen to invade the host, that will cause disease and evade immune defense. So for example, these pathogen have some toxins, so pathogen can secrete some toxin. Pathogen have the enzyme. Pathogen also have some adhesive property that it will adhere to the outer layer of the, let's say, skin. Or it also have some immune invasion mechanism that will come under the virulence factor. So just see here the picture, that adhesion, biofilm formation, hydrolytic enzyme, heat shock protein, thigmotropism, and hyphal production. So these are the some of the factors that can give the pathogen a virulence character. So this will come under the virulence factors. Okay, so coming to the next question. So we do have another disease, and that disease is giardiasis. And the question is, which of the following is likely route of transmission for giardiasis in outdoor setting? And you just need to tell me either the career, or how it is getting spreaded, or how it is transmitted. So the disease is giardiasis. So options are inhaling spores released by rodents. B is drinking untreated stream water contaminated with fecal matters from animal. C is skin contact with tree bark infested with parasite. And D is eating unwashed fruit exposed to sunlight. So you just need to tell me how this giardiasis is transmitted. Yes. So yes, I also agree with the B option, that whenever you will drink untreated stream water, and that if that untreated stream water is contaminated with the fecal matter, that will give you the giardiasis. So just see here the explanation part. So Giardia lamblia is the protozoan parasite that spread through fecal oral transmission, often via contaminated water. And this is how, like in the picture, you can see how this particular protozoa parasite can get infect the food, and how if we will ingest it, or if we will digest it, then how this can give us the infection. Okay, so coming to the next question, which of the following pair is correctly matched with their primarily natural reservoir? So options are like, on the left hand side we have the disease name, and in the right hand side we have the carrier, or the organism that are vector organism. So we have four: plague, botulism, gardiasis, anthrax. And we have four natural reservoir, or you can say carrier or vector, and these are beavers, mosquitoes, beavers, fish. So you need to correctly match the disease with their natural reservoir. So C is the right answer, that natural reservoir for giardiasis is the beavers. So beavers are known reservoir for Giardia lamblia, which can contaminate natural water sources. Plague is primarily carried by wild rodent. Botulism is usually from improperly stored food, and Clostridium botulinum and anthrax is associated with herbivores like cattle and sheep, but not fish. So this is how we can correctly match this C answer. Okay, so coming to the next question. So now we have moved on to the next lecture, in which like we will talk about the photosynthesis. It can be in the presence of oxygen, it can be in the absence of oxygen, it can be uh, like the organic source is carbon, or it can be that the energy source may be inorganic in nature. So we will talk about this. So this question is based on the anoxygenic photosynthesis. So the photosynthesis, but in the absence of oxygen, carried out by green sulfur bacteria. Which of the following is typically released as a byproduct? And the options are molecular oxygen, elemental sulfur, then carbon dioxide, and methane. So yes, if it is a green sulfur bacteria, then the byproduct will be typical sulfur. So just see here, what do you mean by this? So in the photosynthesis, we do have two type, that oxygenic and anoxygenic. In the oxygenic, we have cyanobacteria and eukaryotic phototrophs. But in anoxygenic, we have purple and green sulfur bacteria and purple and green non-sulfur bacteria. And here just see the difference. Then that the electron donor in the oxygenic will be H2O, that is water, but the electron donor will be H2S, H2, or elemental sulfur for the anoxygenic. And the living environment for the oxygenic may be aerobic zone of lakes, river, and ocean. And living environment for anoxygenic will be anaerobic zone and the sediment of lake, river, and oceans. So this is how you can do the categorization between two type of photosynthesis. The one is oxygenic and the another is anoxygenic. And you can just see here that if it is the oxygenic, then we do have the plant does. So plant in the presence of carbon dioxide and in the presence of sunlight and water, this will give you the glucose, oxygen, and water. But if we have electron donor is not water but something else, if it is a H2A and A can be anything, then it will give you a carbohydrate but 2A, and this 2A is not the oxygen. So that's why this is known as anoxygenic photosynthesis. So this is not giving you the oxygen compound, and then it will give the water. And here A can be it can be S, or it can be like this H2A, this can be H2O, this can be H2S, and this can be H2, or any another electron donor. So this is how you can differentiate between the photosynthesis that is either oxygenic or anoxygenic based on the electron donor we have. If the electron donor is oxygen, sorry, water, then this will be come under oxygenic photosynthesis. And if we do not have water as an oxygen for the photosynthesis, then this will come under anoxygenic photosynthesis. Okay, coming to the next question. Which of the following electron donor is used by purple sulfur bacteria in their photosynthetic process? And the option are water, glucose, hydrogen sulfide, and oxygen. We have talked about the electron donor, right? And if we do have a purple sulfur bacteria, then what will be the electron donor for this? So here electron donor will be C, that is the hydrogen sulfide. So this will not be H2O, but this will be H2S. And this is how this will come under an oxygenic photosynthesis. And you can just see here, we have the carbon dioxide, we have the light, and in the presence of H2S, this will give you the carbohydrate, sulfur, and H2O. So thus this sulfur bacterias are phototrophs, are the phototrophs, but are not giving the oxygen that we human needs. So anoxygenic phototrophs do not use water as electron donor, unlike oxygenic phototrophs, for example, plants and cyanobacteria. Instead, they use H2S, which contribute to the production of sulfur instead of oxygen. Okay, coming to the next question. So we already have talked, like we have learned in our primary schools that this is chlorophyll that is responsible for the photosynthesis. But along with that chlorophyll, to increase the efficiency to take the sunlight, we do have another pigment in our plants or in cyanobacteria or in the red algae to take more light. And you have to tell which pigment class primarily assist in light harvesting in cyanobacteria and red algae. And the options are xanthophyll, carotenoid, phycobilins, and the flavonoids. And just see, this is the cyanobacteria and red algae. So this is not A, this is not xanthophyll, but this is phycobilins, because we are talking about the cyanobacteria and red algae. As xanthophyll carotenoids are in like a normal green plant, but if we are talking about cyanobacteria and red algae, so along with the chlorophyll, we do have another pigment that can help us to take light, that can increase the efficiency to take the light, and this can be phycobilins in cyanobacteria and red algae. So just see here, if we have the chlorophyll A and chlorophyll B, these are taking light at a particular wavelength, and it may not be very sufficient, that like the sunlight may not be sufficient at such peak, because we do have light from very short range to very higher wavelength. So to increase the efficiency to absorb the sunlight, we do have another pigment, for example beta carotene and another. And if you can see the peak of this, so this can increase the range of taking the sunlight, and this is how it can increase the efficiency of the plant for the photosynthesis. And in cyanobacteria and red algae, such pigment, one of the such pigment is phycobilin. And these phycobilins are water soluble accessory pigment crucial for capturing light in organism like cyanobacteria and red algae. They transfer the captured light energy to chlorophyll for photosynthesis, especially in low light aquatic environments. Now, the next question is, and yes, again, you can see here in red algae, we have phycobilin, and this is good at absorbing blue light. Okay, the next question is, which of the following correctly contrasts phycobilin and chlorophyll in terms of their cell localization? So you need to tell me the difference between phycobilins and chlorophyll in terms of their cell localization, so where they are present. Uh, so option A is both are embedded in the thalcaloid membrane. B option is phycobilins are lipid soluble, unlike chlorophyll. C is phycobilins are water soluble and not attached to membranes like chlorophyll. And D is chlorophyll float freely in the cytoplasm, unlike phycobilins. So you just have to tell me the difference between phycobilins and chlorophyll in terms of their cell localization. We already have talked, like in last question only, in the explanation it was given that phycobilins are water soluble, right? And these are not attached to membrane, unlike chlorophyll. So chlorophyll are attached to some membrane. If that figure I will have, I will show you. Yes. So just see here. So this is the wall. This is the wall, and in that wall we do have the pigments. So this is the chlorophyll pigment, and the photons will come. So just see here that chlorophyll are attached to the wall. But if we are talking about phycobilins, so phycobilins are not attached, but they are freely moving. So chlorophylls are lipid soluble pigment and that are embedded in thalcaloid membrane, while phycobilins are water soluble and usually part of phycobilisomes. So these are phycobilisomes. Here you can see protein complexes that float on the surface of the membrane. So this is how you can give the right answer to this question. Okay, so the next question is again on phycobilin. So the question is, which cyanobacteria expresses gene for phycobilin synthesis but do not appear to use them for light harvesting. So the question is they do have the gene for phycobilins, but they are not using it. They does not appear to use it for light harvesting. So the question are spirulina, prochlorococcus, and nostoc and anabina. Okay, so let me show you the answer. So this is just like to remember the thing. It is not always that you will remember the things. So this is prochlorococcus. So that have that gene of the phycobilin synthesis, but does not appear to use them for light harvesting. Okay, next coming to the reaction center. So basically, we have talked about the photosynthesis, and for that photosynthesis happen in the chloroplast, right? And in chloroplast, we have pigments, we have some reaction center, and that reaction center is very specific with what wavelength they will take, and based on that wavelength of the sunlight the reaction will move on. So based on that wavelength, so P870 means the photosynthesis will happen when it will take the sunlight of wavelength 870 nanometer. So P870 is the reaction center chlorophyll found in which type of photosynthetic bacteria, and options are cyanobacteria, green sulfur bacteria, purple bacteria, and red algae. So I wanted to tell you that each of the organism have different efficiency, or even you can say different range of taking the sunlight from different wavelength. So you have to tell me when they are taking 870 nanometer wavelength range of sunlight for the photosynthesis, so what kind of photosynthetic bacteria I'm talking about. So out of this four, you are confused between A and B, but the answer is C. So these are the purple bacteria. When the reaction center chlorophyll is P870, and let me show you. Just see here, P870 pigment is specific to purple bacteria, a type of anoxygenic phototroph. It get excites. So just see here. Uh, so first we have this pigment, then sunlight of that particular wavelength, that particular 870 nano will strike. When it will strike, then this center will get excited, and after excitement it will release electron, and this electron will move on to another complexes, and then this complex will give you NAD plus, and then again this will change into the center P870 plus, and this will again change into P80. So there is a kind of cycle. So, if it get excited by light to drive cyclic electron flow, which produces ATP without splitting water or producing oxygen. So with the help of this, they can produce ATP, and you know the ATP is the energy currency for any of the organism. And here we do not need water, or we are not producing oxygen, but without that, energy is being released in purple bacteria. Okay, coming to the next question. Which of the following reaction center pigment is mismatched with the type of photosystem or organism? So you have photosystem 680, 700, 870, 780, and you have photosystem 2, photosystem 1, purple bacteria, green sulfur bacteria, and you need to find out the mismatched reaction center with the photosystem or organism. Okay, just see here. Yes, D is the right answer. Um, we already have talked about this. So, for green sulfur bacteria, the actual reaction center is P840, but not P780. So, that's why D is the right answer. You can just see here, if we have PS2. So PS2 have the range of sunlight 680 nanometer. If this is PS1, photosystem 1, then the wavelength of the sunlight will be 700, right? Okay, so we learned that in the photosynthesis, CO2 is taken along with water and sunlight, but the carbohydrate, oxygen, and water will be released. So, and this is happened in the Kelvin cycle. And you just have to tell how many CO2 molecule are fixed during Kelvin cycle to generate one net G3P molecule. So in the Kelvin cycle, we have to take a lot of CO2 molecules so that we can generate one net G3P molecule. And you have to tell me how many molecules of CO2 we need. Not B, but it should be C. That we need three carbon dioxide molecules to generate one net G3P molecule. So let me show you the this one. Um, so this G3P is glycerylide 3 phosphate, and this is 3 carbon sugar. And if we need 3 carbon sugar, so no doubt we need 3 CO2 so that we can have 3 carbon, right? And let me show you here the figure. So initially we have 3 CO2. So one by one we will take the CO2. So if we have 3 CO2 in the presence of Rubisco, it will generate this um, 5 carbon compound. 1, 2, 3, 4, 5, 6. Initially it is, so initially it was 5, 1, 2, 3, 4, 5. But whenever it will react with the carbon dioxide, it will generate 3 molecule of 6 carbon compound, and that then it will change into 6 compound, but that 6 compound each will have the 3 carbon. And this is how it will take ATP, it will take NADH, and this is how the energy will be utilized. And after that it will give uh, a 3 G3P. So 3 CO2 is giving you just C at the bottom. So 3 CO2 is giving you 1 G3P, and this 3 GP will go on to make the glucose that we need. So based on that, I think you can give the answer. Yeah, this is again the same thing. I think I should skip it. This is again the same Kelvin cycle. Okay, so what is the role of ATP in the Kelvin cycle? To provide reducing power for CO2 fixation, to break down glucose into G3P, to drive the energy demanding step of carbon fixation and regeneration, to excite chlorophyll molecule in photosystem 1. So what do you think? What is the need of ATP in the Kelvin cycle? So, because as I told you, ATP is the energy currency. So, whenever we need the energy to break down some bond or to make some bond, then we need energy, and this energy we can get to break down the ATP into ADP, right? So, yes, C is the right answer to drive the energy demanding step of carbon fixation. As I let me show you here. Just see here, 6 ATP were used, and these ATP were changed into ADP. And again, in next step, 3 ATP were used, and that gave 3 ADP. And these ATP are used because to change the compound or to change any composition, we need energy, and this energy we can get from ATP. So that's why C is the right answer. So ATP provide energy for the conversion of 3-phosphoglycerate into G3P, and also for the regeneration of Rubisco and the CO2 acceptor molecule, both critical part of the Kelvin cycle. Okay, which of the following combination correctly reflect the number of ATP and NADPH used per single turn of Kelvin cycle? So you have to tell how many ATP and NADPH are used for every single turn of the Kelvin cycle. So single turn means one CO2 molecule will be used. So for single CO2 molecule, how many ATP or NADP will be used in the Kelvin cycle? So this is 3 ATP and 2 NADPH are used. So just see here, for each turn of the Kelvin cycle, consume 3 ATP and 2 NADP, and because we need to fix 6 CO2 to produce 1 glucose, right? So for 6 carbon dioxide, or let's say 1 glucose, it uses 18 ATP and 12. You just need to multiply it with 6, right? Because we need 6 CO2. So that's why for 6 CO2 to make 1 glucose, it uses 18 ATP and 12 NADPH in total. So this is the energy intensive process. Okay, next question is again on photosystem. So why does bacteriochlorophyll A has absorption peak in near infrared region? And that is 870 nanometer. And the options are: it helps bacteria compete with algae in absorbing blue light. B is it allow photosynthesis in high light environment. C is it enable photosynthesis in low light and oxygenic environment. And D is it protects cell from UV radiation. Why bacteriophile A have the absorption peak in infrared radiation? As you know that bacteriochlorophyll A, so such kind of bacteria are living not on the top of the river or on the top of the lake, but in the bottom where they do not have much sunlight. But and whatever the sunlight will be present there, that sunlight will be very diffused, will not have higher energy. Not higher energy means that will have the higher wavelength, because higher wavelength means less energy. So when there is the environment, when there is a low light, in that low light they should have the absorption peak where that low light can be also absorbed. So that's why C is the right answer. It enable photosynthesis in low light. Low light means the wavelength will be higher, right? So that's why C is the right answer. And just see here, A and B are having the peak near to 400 and 500 nanometer. But if we are talking about the bacteriochlorophyll A, then that have the peak near to 800 nanometer. And why this is 800 nanometer, you can see the explanation here, that bacteriochlorophyll A absorb light in infrared region to perform photosynthesis where visible light is scarce, such as in deep aquatic or sediment layer. These conditions are typical of anoxygenic photosynthetic bacteria, allowing them to thrive without competition from oxygenic phototrophs like plants and algae. So that's why this is giving me the right answer. Okay, so the next question is, which of the following pigment is least likely to absorb near infrared light? And the options are bacteriochlorophyll A, chlorophyll B, bacteriochlorophyll C, bacteriochlorophyll D. Which pigment will not take infrared light? So this is actually chlorophyll B. So chlorophyll B is no. Chlorophyll B is present in the plants, and plants mostly take sunlight in the visible range, because they do not have the deficiency of the range of the sunlight. They are like they can encounter huge sunlight, and that's why the absorbing peak will be in the visible range but not in the near infrared range. So that's why chlorophyll B is the least likely to absorb near infrared light. So chlorophyll B primarily absorb light in the blue that have the wavelength near to 455 nanometer, and in the red region, not in near infrared. In contrast, bacteria chlorophyll, especially A, C, D, E, G, are adapted to absorb longer wavelength, included near infrared light, helping bacteria survive in light depleted habitats. So that's why B is the right answer. Okay, so next coming to the chemoautotroph organism. So here is the question: which of the following combination correctly matches a chemoautotrophic organism with its typical electron donor? So here the answer will be iron bacteria. Uh, so iron bacteria, here the electron donor is Fe2 plus. So just see here, iron oxidizing bacteria use Fe2 as their electron donor. The next question is, why is CO2 commonly used as a terminal electron acceptor in chemoautotrophic metabolism? As we already have studied that why O2 is the ultimate electron acceptor, because this is abundant and highly electronegative. Whenever there is a highly electronegative, means the combination of this with any substrate. So for example, if this is the glucose and oxygen is reacting with this, and it will give CO2 plus H2O plus energy, and this energy will be high in amount, because O2 is high terminal electron acceptor that have very high electronegative. So just see here, this is the electron tower, and where oxygen is at the top. So this is giving you very high electronegative. So whenever there will be any combination, so this will give you the highest energy ever. Then the next question is based on the nitrosomonas. So which of the following best describe the metabolism of nitromonas? So nitromonas is used to convert ammonia to nitrite. Okay, so here it use ammonia as the energy source, and CO2 as the carbon source. Thank you everyone. I will take, I think, 2 more minutes, and you people can leave, but I will just take 2 or 3 more minutes to complete this. Because I already have talked about nitromonas. So this is used when we need to convert ammonia to nitrite. Then which step of the nitrogen cycle involve nitromonas? So this is the nitrification step, where this ammonia is changed into nitrite. Then we have one more bacteria, Thiobacillus ferredoxin, and it oxidizes ferrous iron to ferric iron, and that help in bioleaching. And if you have heard about the acid rain minage, so this help in acid rain minage. Then they can survive in toxic environment, and because it uses iron and sulfur compound as energy source in acidic pH. And I think 1 or 2 question, and there is a one syndrome that can be happen due to nitrate, and this syndrome is known as methemoglobin, where whenever infant or the child have lower human stomach nitrate to nitrite, and this is how this will give you the some syndrome, and this syndrome is a methemoglobin, and this is uh, mm, uh, so and this methanoglobin is most of the time is misdiagnosed with the disease that is pneumonia. And I think this we can leave. This we can also leave because we already have discussed. This is the last question. Even when saliva is nutrient rich, then even it is not considered as the good microbial culture because it contains enzyme. We already have talked lysosome and lactoperoxidase that will kill the bacteria, and that's how. Thank you so much. Thank you so much for showing the patience. And I think we do have a last class on Sunday, but yes, I also uh, took another extra class so that I can teach 2 more hours. So we do have class on Monday. Then we also have 2 hour class on Wednesday. So Monday and Wednesday we have class from 6 to 8, and that's how the course will be completed. So in the next week, I will cover the content that will be covered in week 12. But on the Wednesday, I will try to cover all the content that we already have covered in a very short way. So this is how we will complete. And thank you so much for showing the patience. If you have any query or the suggestion, you are welcome to give me. You can even ask me question if you have any on my Gmail account. So my Gmail account is Jyoti21, JYOTI 21 @iiserb.ac.in. And if you have any problem, if you have any doubt, and even if you have any suggestion, how can I teach in a better way, you are welcome to suggest me, or you can ask any question. So thank you so much for showing me the patience. We will meet again Monday 6 to 8. Thank you. Thank you so much. Now you people can leave. Thank you, everyone.